support of the Noodleman Foundation, I've been given the opportunity to study in the Brumback Lab, which specializes in autism. And through my experience in the Brumback Lab and reading scientific papers, I've been able to learn about a lot of different neuroscience topics such as autism, optogenetics, and genes linked to autism, and Fragile X syndrome. I've also been able to learn about professionalism in the lab by observing the lab techs who pretty much run everything in the lab, and they have to keep track of all of the mice that are bred and all of the equipment that is ordered. So from watching them, I've been able to learn that organization and cooperation are really big parts of being a professional in the lab. So although I've gotten to observe, I've also gotten to learn about neuroscience through hands-on topics such as brain slicing and genotyping. Genotyping, which is the process of determining differences in the genetic makeup of an individual by examining the individual's DNA sequence and comparing it to another individual's DNA sequence, was one of the techniques I got to do the most. And genotyping can basically be broken down into three different steps, which are lysing tails, PCR, and gel electrophoresis. Before we were able to do any of the steps, the lab techs had to go down to either the vivarium, where the mice are kept, or the Animal Research Center, where some of our other mice are kept, and they would clip the tails for the DNA sample. Then once they came back to the lab, I would lyse the tails, which meant I would isolate the DNA from the tail samples using protein kinase K and direct PCR lysis. Then I would take the samples and incubate them at 50 degrees. After they were incubated, I would move on to the next step, which is PCR. And PCR stands for polymerase chain reaction. And whenever I would do the PCR, I would take the samples and mix them with the master mix and primers, which would target the specific DNA sequence that we needed to amplify. And once I mixed them, I would put them in this little machine over here, which would heat them and cool them at specific rates to amplify the DNA. Once the PCR was done, I would create an agarose gel, which would be kind of a block that's rectangular and semi-similar to silicone, and it would look something along the lines of this with wells in each row. And once I made the gel, I would then take the samples that had gone through PCR, and I would fill them in each of the wells. Doing this step is important for gel electrophoresis because using electricity, we're able to create bands, which are these lines on the gel image right here, and these bands will tell us the genotype of each of the samples once we compare them to the controls. For example, this is an actual picture from some genotyping I did on some SCN1A and SCN1B mice. And so based on the controls, we have the wild type, which is just the first band, and the heterozygous, which should be the two bands up there. So for sample 5 and 6, they both have one band, so I know that they are wild types. And for sample 7 and 8, they have two bands, so I know that they're heterozygous. Another technique I got to do was brain slicing. And brain slicing is a laboratory technique in electrophysiology that allows the study of a synapse or a neural circuit in isolation from the rest of the brain. And this is often done in controlled physiological conditions. And so before we can even slice the brain, we have to extract the brain through perfusions, which I'll go on to explain later. And whenever we extract the brain, then we take it and we usually super glue it to a black platform. And sometimes we will use an agarose solution to go around the base and create a support for the brain. Once we glue it to the brain, we put it inside this machine right here, which is a vibratome. And it has a thin razor on that black block up there, and using the control panel to the side, we can determine the thickness and the angle that we want to slice the brain at. This is very important for seeing the neural activity in the brain, but the technique that you use can change depending on why you need the brain slices. For example, if I needed to look at the brain slices because I had done injections, then I could take up to 2 hours and I would use a different solution than if I was patch clamping, which means that I would need the brain cell tissue to be live, so I would have to go quickly, and I would use different solutions as well. So I've done things, but I've also gotten to see many things, which one of the things I got to see was patch clamping. And patch clamping is a laboratory technique in electrophysiology used to study ionic currents in individual isolated living cells, tissue sections, or patches of cell membrane. Through patch clamping, we can observe single action potentials, theta bursts, and inhibitory and excitatory responses. Through single action potentials, we are able to see how fast and how wide each action potential is and how long it takes for the action potential to hit a resting point. We are able to see this by taking a tiny glass pipette and sticking it into the slice, and we would usually suction onto either a cell or a dendrite, which would be these kind of circles on the image right here, and we would create a tight seal so that we could maintain our access to the cell. Through theta bursts, we are able to take stimulating electrodes and place them on the brain and mimic theta rhythms in the brain to see what the cell activity would be if the brain was functioning at a normal rate. Then another technique that I saw in the patch clamping was the injection of a chemical that is used to block either inhibitory or excitatory responses. And when you do this, you can see the difference between the two responses, but more importantly, when you block the inhibitory responses, it leaves only the excitatory responses to fire, and that can cause little seizures in the brain slice. So by doing that, we're able to see the cell activity if there were to be a seizure in the brain. This is important for Dr. Howard's lab because he focuses in epilepsy. I was also given the opportunity to see a multitude of behavioral experiments. In these behavioral experiments, I actually got the opportunity to create behavioral boxes, and one of the behavioral boxes I helped create was an elevated plus maze. And an elevated plus maze would look actually almost exactly like this diagram right here, and it has the open sides of the maze and the enclosed sides of the maze. And in the maze, you get to test anxiety in mice by placing a mouse in the middle and seeing whether it explores the open sides, which is an activity that a typically developing mouse would essentially do, or whether it stays in the enclosed parts of the maze, which would be a more anxious behavior. I was also able to observe a 3 chambers test, which looks like this one here, only except in our lab, our 3 chambers were white walls, not transparent clear walls. And in this test, you are able to test social interaction in the mice by placing a mouse in the middle and opening one door so that it can interact with the familiar mouse, and then you open the other door so that it can interact with the new mouse. And this tests social interaction by seeing whether or not the mouse would rather interact with a familiar mouse or a new mouse, which could be a good model for autism, since autism can cause social interaction deficit. I was also able to see an open fields test, which is just a big plastic box, and you put the mouse in it to test locomotion and anxiety. And you can test anxiety by seeing the movement of the mouse. For example, if the mouse were to stick closer to the walls, that would be a more anxious behavior. But if it went to the middle, that would be more typically developmental behavior. And through this, you videotape all of the experiments that I've explained, and once we videotape them, then an undergraduate showed me how we video score them, which means that we look through at the videos and we note specific behaviors that need to be noted based on the type of experiment that the video was of. And the third thing I got to see, which is also my favorite, were the mice surgeries. And I got to see perfusions and injections. And perfusions it's a procedure that is done so that you can extract the brain. And in order to do this, you have to drain the body of all of its blood. Because if you were to extract the brain without doing perfusions, the blood toxins can mix with the brain, and then you would no longer have a viable piece of, a viable specimen. And in perfusions, you have to cut at the diaphragm and cut up, and then make an incision on the heart and stick a needle through the left ventricle, and then using chemicals such as PBS and formaldehyde, you push those chemicals through the body and push out the blood toxins. You know that this is working whenever the liver, whenever the other organs start to turn to a pinkish color. The second mice surgery I was able to see were injections, which would look along the lines of this diagram over here. And injections are a procedure that is done when color tracers are injected into the brain to check placement or to see neuronal activity. And you do this by first putting the mice under isoflurane, which is an anesthetic, and then you have to shave their heads and make an incision so that you can gain access to the place that you want to the skull. And once you gain access, it's important that you use either a brain atlas or bregma lambda, which are two biological markers in the brain, to find your coordinates that you need for targeting the right part of the brain. Then once you make the injections, you suture up the mice and put it back in its cage for recovery. Once you do these injections, you can do brain slicing to see where the path of the dye traveled, to see the neuronal activity and what neurons lead to where. And what I've enjoyed. So I've enjoyed everything that I've gotten to do here this summer, but most importantly, I've enjoyed the conversations I've gotten to have with people in the lab. Through these conversations, I was able to learn about everybody's background and all of their lab experiences in the past. And I was also able to learn about their college experiences, which are really helpful to me as a student as I venture down that path soon. What I valued the most were the conversations I got to have with my mentor, Audrey, because through those conversations, I was able to ask her any and all questions I had about neuroscience and her profession, and she's just a really great role model for young women in science. Okay, tearing up a bit. Okay, sorry. What I will carry with me. That is kind of embarrassing.
It means it was meaningful. It means it was meaningful.
I'm gonna start crying too.
I know. Oh my god, okay.
I think
So this is a mess.
you're okay. Deep breath.
Ooh. I did not do my makeup to cry it off. Okay. What I will carry with me, I will carry a lot of physical things, such as my student CV and and my laboratory journal. But I will also be able to carry the relationships and memories I've gotten to make, and I will be able to leave with a newfound confidence in my future in neuroscience. So thank you so much.